Welcome back, ladies and gentlemen. We are continuing our discussion today on your adaptive immune system or these targeted defenses in your body. And so let's continue talking about how your immunity actually gets built. And so we're going to start today by talking about this idea of antigen and antibody. Because it really is the hallmark of how your body is able to learn a particular pathogen and then remember it in the future. So when we look at antigens, right, typically we think of these as foreign molecules that are going to produce some sort of immune response, and thus they are things that are found on the surface of pathogens. These are proteins, carbohydrates, or some combination thereof typically. That being said, you do have antigens on your own cells. They're basically just tags. They're identification tags. Sort of think of it like wearing a name tag in a room full of people you don't know. And it tells you a little bit about them as well. So, what their job is, perhaps, or their career. It's kind of what these antigens are. They allow our body to be able to recognize what everything is when it comes in contact with them. And then the antibodies are proteins that your body will create that are correspondingly shaped to a particular antigen. So it's sort of like a lock and key idea. All right, so when we come across a particular antigen shape, your body then produces antibodies that are correlating to that particular protein complex shape. So that those antibodies then are able to recognize a specific antigen and then tag it and communicate with your immune system to say, hey, this is this antigen that I just found. And do we have memory of it? Well, probably because we've produced these antibodies at this point. You may also come across the term immunoglobulin, or you see this shorthand IG. That's just another way to talk about an antibody. They're interchangeable terms. Okay. So again, antibodies contain those antigen receptor connection sites. Right, these antigen binding sites here on the ends. They're correspondingly shaped to the antigen that they connect to. So just a couple of terms here, right? So here is the antigen itself that may be on the pathogen or just any cell for that matter. And then it really is the epitope. This is the binding site where those antibodies are then correspondingly shaped. So that is what allows us to have such high specificity because we have that bonding site to those antibodies that correspondingly shapes to that epitope binding site on the antigen itself. So, speaking of antibodies, there's actually a huge class of them. Antibodies aren't all created equal. There are different ones that we create that are different sizes, different shapes, come from different places, do slightly different functions. Um, so, I just wanted to kind of point that out to you here. We typically use the IG designation and then they get a letter corresponding to what that is. So when we look at something like the IgD antibodies, these are monomers, meaning they're the single sort of Y shapes that you're typically used to seeing. And these will specifically attach to the surface of B cells. So this is really important in activating B cell differentiation. And we'll talk a little bit about how that activation happens a little later. IgMs, these guys are pentamers, meaning there's 5 of those Ys put together, and I'll show you a table in just a second of what this looks like. But these guys get released by plasma cells. Remember, those are activated B cells that are functionally producing antibodies, and so they are typically utilized or produced during your primary immune response. What does that mean? If that's a new term to you, primary immune response basically just equates to the first time you've ever encountered a particular pathogen. So it's the first time you've ever gotten sick with something. And so these are the antibodies that are being produced by that first encounter. IgGs, These are, again, monomers, so just that sort of single Y structure. These are the most abundant and diverse antibodies in your body system. They can get produced and we can create lots of them, both during your primary immune response, or the first time you come across a disease, as well as subsequent exposures, which would be that secondary response. These are also really important in the sense that they can cross the placenta, meaning that mom can pass these to baby before baby is born. And so they are very tightly connected to what we'll talk about as passive immunity. And one version of passive immunity is mom being able to take all of the antibodies that she has produced from all of the exposure to the pathogens she has encountered in her life and be able to pass those antibodies across the placenta to baby before baby is born, so that when baby is born and baby can't produce their own antibodies yet, their immune system isn't developed enough at birth to do that, that these antibodies hang around in baby's blood and body for a few months to be able to give that baby, I should say, some protection until the immune response in that baby is strong enough to be able to start producing their own antibodies and provide their own body some protection. IgAs, these are dimers, so two Ys connected. These help to prevent the attachment of pathogens specifically to epithelial cell surfaces. So a lot of your skin cells would be epithelial, but generally we're talking inside your body, so things that kind of line all of your organs would be where these guys might be functionally active. These can also pass or confer passive immunity, but now instead of going from mom to baby before baby is born, IgAs are small enough to be able to pass from mother to infant during breastfeeding. So mom will pass on again a bunch of her antibodies as she makes this connection with baby. Baby actually, when baby suckles, some of that saliva actually ends up back into mom's breast tissue. And so her immune system is monitoring what sort of things might be in baby's saliva. And if it picks up on a antigen from a pathogen in baby's saliva that's now in her breast tissue, that she has immunity for, her body will sort of upregulate and create a bunch of antibodies against that pathogen and then pass it back to baby through the breast milk. It's kind of a pretty crazy feedback system, which is pretty cool. Um, IgEs, these are monomers, so again, single Y structure that are going to bind to mast cells and basophils, which causes the histamine to get released. So these guys are a connection to your inflammatory response. So this table just does a nice job kind of taking all of that information and giving you a little bit of extra background. It also gives you some of those different structural components. So these would be your monomer structures, just the typical Ys. Here's that big pentamer, and then here are those dimers. So kind of cool. Each of these antigens is a marker on the surface of a particular cell or particular pathogen, and then your body will produce specific antibodies shaped to those antigens. So the production of those antigen binding sites, we'll talk a little later, is genetically controlled. And it's kind of interesting how we use a set of genes to really be able to create this huge diversity, this huge variety in markers. But for now, that's the way this works. So we use those different created antibody binding sites, antigen binding sites, to be able to target and recognize specific invaders. So keep in mind that those antibodies aren't directly attacking or directly destroying those pathogens. Instead, they're just tags. So when those antibodies find their corresponding antigen, they will bind to it, and then that will elicit an additional set of immune responses to ultimately destroy that pathogen or destroy that particular molecule or cell, whatever the connection happens to be. So they trigger additional mechanisms that neutralize or destroy those invaders. They themselves are not directly destroying the pathogens. They're just tagging mechanisms to say, hey, this shouldn't be here, and then alert your B and T cells to finish the job. So when we look at that connection between antibody and antigen, once that complex is formed, we sort of go through this process: neutralization, agglutination, precipitation, and then amplification of some additional non-specific responses um, from phagocytosis, cytolysis, so just sort of blowing up cells, and then inflammation as well. So when we look at this, right, once that antigen and antibody find each other, remember that this is part of that humoral immunity. So they are going to complement each other. They're going to work together. They're going to activate that complement system, which can enhance things like these nonspecific defenses of phagocytosis, so bringing in more macrophages and things like that to kind of eat things up. It will elicit some additional inflammation, which can be localized, or it can be fever, and so systemic. We can also cause cell lysis, whether that cell lyse being destroyed is a bacterial cell itself, or if it is a cell in your body that has been turned into a virus factory. Um, they are going to neutralize them, so they will allow the binding of them to block viruses, for example, from continuing to be able to get to receptors to get into your cells. If they are some sort of toxin produced by bacteria, they can prevent those toxins from continuing to attach to receptors and things in your body and causing problems. They can cause agglutination, so they will cause cells to bind together and stick together, and if they're in this massive clump, they can't really get to reproduction if they're bacterial cells, or if they're parts of your body, just sort of inactivating them in a giant clump, causing them to also precipitate out in solution. So that's the idea there. Interesting thing is that when we talk about vaccinations, um, up to this point, what we are ultimately trying to do when we create those vaccinations is elicit your body to create antibodies for that particular antigen. And we can do that in a whole host of ways when we build those vaccines. But the ultimate goal is to ensure that that antigen, or at least some subset of antigens on that pathogen, are presented to your immune system in a way that allows your body to build antibodies against it without actually causing you to get sick. The tricky part here is that building those vaccines, I need to know which antigens elicit a strong enough immune response from your body to allow your body to continue to remember. And this is a lot of times trial and error on our part. We still don't fully understand how and why these work. So let's just take the flu vaccine, for example. So a lot of people are like, well, I got the flu vaccine, but I still got the flu. There's more to it. And the reason being is that, first off, there are multiple strains of the flu. Two, the flu itself collectively, as a class of viruses, are really good at mutating. And so we are producing flu vaccines months and months before flu season ever hits, with the hope that we're choosing the right strains for that particular season. Sometimes we do a really good job, sometimes not so much, and sometimes the virus just mutates in the meantime, and we're not able to ramp up vaccine production fast enough to accommodate that, because it definitely takes months, sometimes even up to a year, to get those vaccines up to production enough to have enough doses to cover the population. So one of the things that's out there in research right now is, instead of driving at creating vaccines against flu strains that specifically go against those epitopes, right, the binding sites at the very tips of those antigens, if we can create antibodies instead and vaccines instead against these spike pieces of that antigen, that piece seems to be much less likely to mutate from season to season, and it's much more universal across different strains of flu as well. So less mutation, and it's more consistent from different strains. If we can manage to elicit an immune response against that portion of the antigen as opposed to the epitope tip of it, then it's possible for us to create a universal flu vaccine, which would be really, really cool. Lots of places are working on that research, and we'll just have to kind of sit back and watch and see if we can get it to actually function.